Hi everyone, today I'm going to introduce my project, the stochastic pulse generator. The project is presented by Yujia Liu on December 11th, 2018. First, I would like you to recall that gene expression has a lot of noise. From the first repressor implemented in 2000, researchers have been interested in the origin of these nasty noises that impair genetic circuits, among which the transcriptional burst hypothesis suggests that the activity of RNA polymerase is not continuous, but subject to stochastic initiation. Translational bursts are observed later as well. Noises are commonly considered as nuisance in synthetic biology. However, recent studies have shown noises could even be functional in several ways. For example, in bacterial stress sensing, the response depends on the frequency of stochastic pulses of upstream protein instead of its absolute concentration. Here we propose a synthetic circuit that generates stochastic pulses, sampling the noise that arises from transcriptional bursts. The circuit proposed here features randomness in intervals between pulses and their strengths, and thus differs from oscillators. The fate of both off toggle switch is determined by the noise of expression. This would be a feasible way to amplify transcriptional bursts and pulse the burst state for long enough. Enlightened by such previous works, a both off toggle switch is used as a noise source in our circuit. The strong constitutive promoter from IgM repository is used here to make leaky expression. If the promoters are too tightly repressed, then there will not be any noise. Also, degradation tags are put after leg I and head R to limit the existence time of these repressors, and act as a redundant design to increase the stability of the system. The noise generated by leaky toggle switch is then fed to a rising edge detector on the bottom branch. The circuit is composed of one direct positive feedback and one negative feedback with delay. When the leaky switch is changed to an on state, the circuit first expresses lux R to activate itself. Then the activation is repressed by lambda repressor with delay. Thus a pulse is occurred. The pulse compositor framework does not apply to stochastic models. Here we demonstrate a discrete time model to simulate the system. The system is simulated in time steps. The production of mRNA in each time step subject to probability distribution. Conventional continuous models do not capture the transcription process in a single cell. This is due to several reasons. Transcription machinery only exists in several copies, and transcription is very rare due to aborted initiation. Thus, stochastic models are demanded to analyze the system. As is reviewed in previous work, the transcription in single cell subjects to Poisson distribution. In more detail, the number of mRNA produced in a certain period of time obeys to Poisson distribution, and the average number of mRNA produced is proportional to the fraction of activated genes, which is given by Hill function, the same as in continuous models. Consider the general gene expression models as follows, and we make two assumptions. First, binding of RNA polymerase or ribosomes are neglected, and both RNAP and ribosome are sufficient in cell. Second, only transcription is stochastic. Binding of activator and translation are still considered as continuous processes. Keep these assumptions in mind, we can get concise but powerful equations depicting stochastic gene expression as follows. To demonstrate the idea, the stochastic expression of a constitutive activator regulated gene can be described as follows. Notice that the parameter here in Poisson distribution is the same as the rate expression in continuous models. We use such model to simulate our system. Here are the results. First, we can see that the both off toggle switch competes between on and off state. The mRNA level is discrete instead of a smooth curve as is shown in protein level. The on state is represented by the concentration of the repressor leg I as shown in blue line. From 0 to around 12,000 seconds, we can say that the toggle switch generally remains on, The system has been stalemated for around 3,000 seconds, then it goes to an off state for 5,000 seconds from 15,000 to 20,000 seconds. The system behaves similarly to the end of the simulation, on and off competes, and alternately wins. The concentration of LAX-R resembles LAC-I, since one of LAX-R gene is in the same operon with LAC-I. LAX-R is the activator that induces GFP. At 0, 8, 26, and 34,000 seconds, there is rather significant rising in LAX-R concentration. There are corresponding pulses of GFP shown in the bottom diagram, which indicates the system works as expected. There are also smaller pulses as a result of stochastic transcription of GFP. The pulses are of different strengths, and the intervals are random. Now let's play around a little bit with parameters. When the basal expression is dialed down, the system maintains the on state. Since the on and off states are symmetric, the system can also go off state. However, the target is not exactly symmetric because the on state drives retroactivity, while the off state does not, which means the off state has higher probability to rule. Nevertheless, the on state is observed here. The system is very sensitive to basal expression, and the low basal expression causes the system to be stuck in only one state. That's all for my project. Thank you for listening.